The overall goal of the following experiment is to assess acute axonal degeneration mechanisms in real time. This is achieved by isolating Thy1 YFP-positive murine spinal cord tissue while continually perfusing the tissue with oxygenated low-calcium artificial cerebral spinal fluid. Then, the spinal cord tissue is aligned under a two-photon microscope objective while maintaining physiological conditions, and the myelin is stained with the lipophilic fluorescent dye Nile red. Next, a micro-sized lesion is created on the dorsal surface of a dorsal column by adjusting the laser power in order to transect myelinated axons within the columns. The results show retraction of the damaged axons, periaxonal myelin swelling, and other cellular responses based on quantitative measurements of the images.
So the main advantage of this technique over existing methods is that we use the whole cervical spinal cord, and therefore the dorsal column fibers remain intact. So this avoids damage to the underlying central myelinated fibers, which commonly occurs during white matter strip preparation. Demonstrating this procedure will be Dr. Starlyn Okada, a postdoctoral fellow in my laboratory who really championed this technique.
To prepare for the intracardiac perfusion, make low-calcium artificial cerebral spinal fluid, or ACSF, with 2 times concentrated stock B and C solutions. For perfusion of the ex vivo spinal cord,